Welcome to your Biocast. I'm Mrs. Jaeger and I'll be your guide. As always, take detailed notes and don't forget to bring them to class the next day. Take some time right now to get your binder together, a pen, and get ready to learn more about biology. Hi, and welcome to your second Biocast video. This day's lesson is going to be about the scientific method. We're going to just kind of review the scientific method and how we're going to use it in class. So, who are scientists? We are all scientists. Anytime you attempt to solve a problem and research a problem and figure out a solution to a problem, you're being a scientist. The picture that you're looking at kind of demonstrates the typical view of a scientist. That would be the guy on the left of the screen holding the beaker. And then we realize that anyone who is exploring the natural world, whether that's through dissections, as you see on the right side, or exploring nature, as you see with the turtle, that is being a scientist. So you guys are scientists, not just people in labs. The first step is to make observations. This would be a good time to take notes. I hope that you're writing this down. When you're making observations, you want to make sure that you are objective and you're not just using your own opinion. Objective means you're taking in all of the information you can, you're using your senses, and you are being not bringing your opinion into the situation. You can use inferences. An inference is a logical interpretation of data based on your prior knowledge. An example of an inference would be if you are sitting watching your television, if I can observe that you are sitting watching your television, I can infer that you like the program because you're watching it. So it's more of a guess based on your prior knowledge. Anytime you see a word in red during a biocast, that's one of your vocabulary words. So if you've not written down the word inference, you want to take some time to do that now. All right, so throughout this biocast, we're going to look at an example of a situation where we'll use the scientific method. Let's say that you have a toaster and you notice that it's not working properly. You put the bread in, no toast comes out. That's a problem. That's an observation that you've made. And the next thing that you're going to do in the scientific method, step 2, is to wonder and ask questions. So you will probably wonder, why is the toaster broken? What's wrong with the toaster? Your question has to be testable. It cannot be something like, are there extraterrestrials? Because we can't test that. We can, however, test and find out what's wrong with your toaster. Step 3 is to form a hypothesis. Notice the red word. What does that mean? Hypothesis is a prediction of what you think will happen if you test your problem, okay? It's also known as an educated guess. You'll see that probably on the OGT. Remember, this hypothesis has to be testable, and it's often written in an if-then statement. So if the toaster is unplugged, then it wouldn't work. Okay, does that make sense? All right, we'll look at it. Next slide. Step 3, so you're forming your hypotheses. Hypotheses is the plural of hypothesis, by the way. You may have 2. Your first might be the toaster is unplugged. So your hypothesis could say, if we plug the toaster in, then it will work. Okay. Your second hypothesis could be that the heating unit maybe just isn't working anymore and it's burned out. Both of those we can test, right? So to test them, you need to set up a controlled experiment. An experiment is just a procedure that tests a hypothesis by collecting information or data. We want to control the conditions that happens under. Okay, we'll talk about control in a little bit. A procedure is just a set of detailed steps used to conduct an experiment. Your procedure should be numbered. It should be very detailed so that someone else could read your procedure and follow it and replicate it. Okay, so for our example, our experiment could be to check the plug, in which we'd have to write a procedure for that, right? Plug it in, see if it works, so-and-so. The second one could be to take the toaster apart and test it. Now, you wouldn't just want to take your toaster apart without knowing anything about how toasters work. So you may need to do research about your topic first. Okay. We'll also collect data in this part of the experiment. In class, you'll be collecting a lot of number data, numerical data. For this example, you'd probably just look and see if it works. That's collecting data. There are two types of data we'll look at. That is quantitative data and qualitative data. Quantitative data sounds like quantity, right? Quantity refers to a number of things, numbers. So quantitative data is data expressed as numbers. This can be things like plant height, temperature, volume, mass, things that are expressed in numbers, data that's expressed in numbers. The second type of data is qualitative data. It describes the quality of something. It describes the characteristics of something. So this could be the color of leaves of a plant. It could be how you're feeling, things like that. So you should know the difference between quantitative and qualitative data. Tables and graphs will be a large part of the data that we collect in this class. Step 5, analyze. You need to reflect on your results. You need to go back and look at what the results are telling you. The analysis should tell in general what you observed. This will be usually expressed as a graph and one statement that explains the graph. Step 6, form your conclusion. For our example, if the toaster was unplugged, and once you plugged it in, it started working, we would say our first hypothesis was supported. If the wires were broken, you might say that your second hypothesis was supported. A lot of times, neither or none of your hypotheses will be supported, which means you have to start the cycle all over again and come up with a new hypothesis. With your conclusions, you want to evaluate your hypothesis. You have to go back to that hypothesis and explain, was it supported or accepted, or was it not supported or rejected? And usually your conclusion is written in a paragraph form. Okay, last slide of the day. There are several variables within an experiment. We're going to talk about these a lot. You're going to need to know how to identify all of these in an experiment, so make sure you're writing these down. We have two groups that we test on. We have a control group and an experimental group. Now, for our purposes, let's think of another example. Say you want to figure out whether or not plants can survive if you water them with Gatorade, okay? Your control group would be a plant that is not watered with Gatorade. Your experimental group would be the group that is manipulated, meaning you're testing it. So you would actually be pouring the Gatorade on the experimental group. Okay? There are two types of variables in an experiment, the independent and the dependent variable. In the independent variable, this is what the scientist is changing. Okay? So instead of using water, we're using, Gatorade. So your independent variable in that example would be Gatorade. Okay? The dependent variable changes depending on whether or not the independent variable works. So for our example, what would change? I want you to take a minute and think about what would change about a plant if your independent variable, the Gatorade, works or doesn't work. Okay, you might notice some differences in plant color, plant height. Those would be dependent variables. Okay, we're going to conclude in just a minute, but before we conclude, I want you to think about a time when you were a scientist. Think about a problem that you solved, or maybe didn't solve, but that you tested. And until next time, I will see you for Biocast 3 next time. Have a good day.